Good morning. I want to thank everybody for joining us in person as well as virtually. My name is Leslie Fesher, and I'm one of the medical oncologists here. And it is my pleasure to introduce Dr. Jennifer McQuade, one of our colleagues at MD Anderson. She is a melanoma medical oncologist, and she's going to be talking about something near and dear, I think, to everybody: diet, microbiome, and roles in immunotherapy.
Great. Thank you so much, Dr. Fetcher, and to the AIM team and the Michigan team for inviting me to speak today. And I'm really sorry that I was not able to be there in person. It would be lovely. Although I will say in Texas, we do actually finally have some decent weather today. And so, as Dr. Fetcher said, today I'm going to be speaking about the role of microbiome and diet in melanoma. I'm an assistant professor in melanoma medical oncology at MD Anderson Cancer Center, and I actually did my my medical residency at Penn, which is where Dr. Fetcher was from before, and so actually learned about melanoma initially from Dr. Fetcher, but this was back in the day before we had all these nice effective therapies. So these are my disclosures. Okay, hopefully that's correct now. So as we've been hearing, outcomes with metastatic melanoma have really improved dramatically, but are heterogeneous, again, meaning not all of our patients will have response to these novel therapies. And some patients, we have the same diagnosis, same treatment. Some patients it benefits. Some patients, but they experience some of these immune toxicities. Others, it's beneficial and not toxic, which is, of course, the best bucket. Some people have not beneficial but toxic, and of course, some have not beneficial as well as not toxic. So how do we identify who are these patients who are going to respond and whether or not they're going to have toxicity? Now, when we say that a patient's tumor either will or will not respond, then it's about a 50-50 chance of whether the tumor will respond to immunotherapy. One of the first questions that my patients always ask is, doc, is there anything else that I can be doing? And while there's been a ton of research looking at the role of lifestyle factors such as diet and exercise on cancer risk, not really melanoma specifically, but on cancer risk in general, what there really hasn't been great data on is, do these lifestyle factors actually influence outcomes for patients that already have established cancer? And so a lot of people say, why is my doctor not talking to me about these things? And it's because there's really actually been a lack of good research. And if you look at the recommendations around lifestyle and cancer, it's really around cancer prevention for this reason. And the guidance is, after a cancer diagnosis, follow our recommendations if you can. Now, we wanted to actually dive deeper into this and actually ask that question, do lifestyle factors influence melanoma biology? Do they influence response to immune and targeted therapy? And so usually when we think about what are the factors that determine immunotherapy response versus resistance, we look at pathways within the tumor. We look at, is the tumor hot with lots of immune cells infiltrating, or is it cold? But one dimension that really was not being well studied was, what about the role of our overall systemic immune tone? And particularly these factors, what we call host factors, that might be able to impact this. So this word host basically means the person that the tumor is residing in. Now, I know nobody here wants to host a tumor, but the idea here is, are there things about us as individuals that might impact the biology and the response of the actual cancer? And so we think about some things that we cannot modify, such as age and biological sex. Of course, from our recent experience with COVID, we do know, right, that these are things that can be associated with differences in both risk as well as outcomes with COVID. And then again, coming back to, what my patients ask, which is, things that they might be able to change. And so things such as obesity, diet, exercise, stress, and now we do have some of this data emerging for each of these, still at very early stages. Now, one of the things that's made this all really interesting and exciting, and I think caught the attention of the scientific community, is this emerging role for the microbiome. And I'll tell you more what that is. But the microbiome, all these bugs that live in us and on us, all these bacteria that are actually really nice integrators of all of these different factors and have been shown to influence response to immunotherapy. So what is the microbiome? So again, we all have trillions of microbes, bacteria, fungi, viruses that live on us and in us. And a lot of these are in a beneficial symbiotic relationship. They make up actually 3% of the human body mass. There are more microbial cells than there are human cells on us. There are more microbial genes than human genes. And the largest number of these microbes actually live within our gastrointestinal tract, where they help break down medications, they help break down food, and they also work with our mucosal immune system and train our overall immune system. Now, the human microbiome has been well studied in the context of many diseases. So metabolic diseases such as obesity and diabetes, also autoimmune or inflammatory diseases such as eczema, allergies, asthma, gastrointestinal diseases, which of course makes sense given where these microbes are, so inflammatory bowel disease. And then there also appears to be a kind of brain-gut axis. So even looking at psychological disorders. So within the field of immunotherapy, this came to the forefront back in 2015 when there were a couple of studies that were published in a journal called Science, showing that the gut microbiome could influence response to immunotherapy in mice. And so what this experiment showed on the bottom, so SPF, so those are the mice that we usually work with in the lab. We keep everything clean within those lab spaces, but they do still have bacteria that live in our gut, just like in their guts, just like we do. And so that's called specific pathogen free. Now, you can also have mice where from the very beginning, there's no microbes. And so they are germ-free mice. Now, if you give a mouse immunotherapy that is an SPF mouse, you can treat their cancer with the immunotherapy and the tumor shrinks, which is over there on the left. But now if you repeat that same experiment in germ-free mice, so they do not have any bugs living in their gut, then we don't have a response to immunotherapy. And so lab mice are, of course, much simpler creatures than we as humans are. And so the question became, is this relevant actually in humans? And so there have been many, many publications now basically showing that if you collect fecal microbiota specimens from patients as they're starting immunotherapy, then there are certain bugs within the gut that tend to be associated with improved response to immunotherapy. Now, this is, of course, correlation and not causation, meaning they're associated, but we don't necessarily know that those bacteria actually had any mechanistic impact on immunotherapy response. And so now to look at mechanisms, we come back to mouse models, and that's how we prove cause and effect. And so here, what they did is they took the fecal specimens from humans who either responded or did not respond to immunotherapy, did a fecal microbiota transplant from the human into the mouse, implanted the mice with tumors, and then treated them with immunotherapy. And so what we see is the non-responders, so the mice that received the fecal specimens from non-responding humans, their tumors also did not respond to immunotherapy. So that's the NR, and we see bigger tumors, higher tumor growth. Whereas those mice that received the responder microbiota specimens, their tumors also responded to immunotherapy. And so those are these in blue. And so we see that we can transmit responsiveness to immunotherapy via fecal microbiota transplant, again, in mice, much simpler creatures. And so this brings us to the fact that the microbiome is exciting because it is both a a biological kind of signature predictor of response, but it's also potentially modifiable. And so it's a target as well. And so how can we modulate the gut microbiome to enhance response to immunotherapy? Well, we can do what we did in mice, and I'll show you some data around that, actually, of doing fecal microbiota transplants. We can give specific bacteria. So we'll talk some about probiotics, but also kind of this next generation of targeted probiotics, which we call microbial consortia. So many beneficial bacteria together in kind of a mini ecosystem. And we also know that our diet is a major driver of our overall gut microbiome. And then what about, instead of trying to increase the beneficial bacteria, what if we actually try to target and decrease bad bacteria? So in terms of the fecal microbiota transplant, there have actually been now 2 very early phase studies where they took fecal specimens from, again, patients who had actually had a response to immunotherapy. And they then took those specimens and gave them to patients whose tumors had not responded to immunotherapy. And they didn't just give them the fecal microbiota transplant, they also then restarted immunotherapy. And so what we see is that in each of these studies, again, these are super, super small, 12 to 15 patients. What we see is that we see some patients where this fecal microbiota transplant is actually now able to allow these patients to respond to immunotherapy on the order of about 30% of patients. Again, small studies, and this needs additional studies to prove this. But fecal microbiota transplants have also been used actually to treat immunotherapy toxicity. And so here, this is a colonoscopy picture from a patient that had colitis. And so this bottom one, this post-FMT, that's what it should look like, nice and healthy. This is a patient that had actually already had 3 different immunosuppressive drugs to try to calm down that colitis, and it was still refractory. And so they did a fecal microbiota transplant. And what we can see is that they got rid of all of that inflammation within the colon. Now, again, this is still very experimental, but kind of an interesting proof of principle. Now, again, the gut microbiome is also exciting to us because it is something that we can control to a greater degree than other genomes, right? So we essentially have 3 genomes. We have our germline, which is, the cards that we were dealt by our mom and dad. We have the tumor genome, which we, of course, don't want to try to change. We just want to get rid of that tumor. And then we have this microbial genome. And this is actually only about 10% genetically determined and is mostly actually determined by our environmental exposures and by things that we consume, so medications as well as our diet. So I will have much more in common of my microbiome with, say, my fiance who lives in the same house, eats the same food, as opposed to my brother who is actually a farmer in rural South Carolina. So we started investigating the impact of diet on both the microbiome and immunotherapy. And so we did this by, when we have our fecal collection protocol for our patients that are starting immunotherapy, we integrated a lifestyle questionnaire. And we first actually really focused on dietary fiber, because a lot of the bacteria that have been associated with response to immunotherapy are bacteria that we know have key roles in fiber fermentation. So fiber from plants and legumes, vegetables, fruits, whole grains, all of these things are actually otherwise indigestible to us if it is not for these bugs that live in our gut. When they break down the fiber, they release these metabolites called short-chain fatty acids. And these short-chain fatty acids are actually the primary nutrient source for these cells that line our gut. And they help us then produce this nice, thick mucosal barrier that leads to an overall kind of better gut health. And so then we studied this in humans, and then we also studied this in mice. And so what we see is that our patients that are habitually consuming the recommended amount of fiber of at least 20 grams of fiber per day, they actually have improved outcomes with immunotherapy. Now, again, this is habitual diet, right? This is not a dietary intervention. This is just asking patients what they usually eat. And there are a lot of other things that these patients might have also been doing that are different from those with insufficient fiber intake, right? So maybe it's a matter of they were feeling better, so they were able to eat a better diet. They may also be the ones that are exercising. Socioeconomic status may play a role as well in determining, whether you have access to all of these fruits and vegetables or whether you live in a food desert. So again, now we come back to a mouse model. And within the mouse model now, we take mice and we treat them either with immunotherapy or with the control. So that's that ISO control. And then we feed them either a high fiber diet or a low fiber diet. And so what we see is in the dark green, the high fiber fed mice treated with immunotherapy have a beautiful response to immunotherapy. Whereas the low fiber fed mice, so now this is the light green, we see a much more attenuated, a much decreased response to immunotherapy in those mice. So again, now we have habitual diet, and then we have diet modification in mice. And now the question is, could we intervene on the human microbiome through dietary interventions? And so there is evidence indeed that we can do this. And so this was actually a really cool study that was published a number of years ago, where they took African Americans in Pittsburgh eating a very prototypical kind of Western diet, high, animal proteins and saturated fat, or Northern Africans with a very agrarian plant-based diet. And they swapped their diets for 2 weeks. It's called a controlled feeding study. So they're giving them all of their food. And what we see over here on the right is that in the Northern Africans, as the fiber content in their diet goes down, so too does the capacity to produce these short chain fatty acids. So the fiber fermentation bacteria go down. In the African Americans, it goes up. And then we see the exact opposite in terms of the bile acid deconjugation. And so those are the things that are involved in animal protein and fat metabolism. So now, can we do this in the context of cancer and of immunotherapy? And so we actually took this same approach. And we have a research kitchen at MD Anderson where we actually are cooking patients all of their meals, and we provide all of the food that a patient takes in over the course of this study. And I'll show you some data. We first did this in melanoma survivors just to show that we could do this and people would eat the diet. And now we're doing it in the context of patients actively receiving immunotherapy. And if we can do this, ultimately, of course, we can't feed the world. And so now the idea is, can we move into interventions where we're actually able to help people change their behavior and then move it, to kind of a multi-institutional setting. And so patients are able to do this. So this is the 10 patients in our initial survivorship study. So we start people at 30, go to 40, and then up to 50 grams of fiber per day. Everybody's able to get up to 40 to 50 grams of fiber. However, we just gave people food. We didn't teach them. And what we find is that if we don't teach people after this dietary intervention completes, then everybody goes back to eating their same usual diet, even though they said this was great, I loved it, and I want to keep doing it. So we need to help people with behavior change. There's some minor bloating and kind of early satiety. None of these are clinically significant scores, and we work with people to kind of mitigate that. And then what we see in terms of the microbiome is that we shift towards a more kind of connected ecosystem and a greater role of the fiber responsive bacteria. And then we also see changes within the metabolome, so things that are produced either by the bacteria or by the host upon consuming. And specifically, we actually see a decrease in a lot of really nasty inflammatory fats and metabolites. Now, are there other ways to modulate the microbiome? This is a study of looking at fermented foods, so things like kimchi and sauerkraut and kefir. And this was in healthy populations. And what they see is that when you eat a lot of those foods, you actually increase the overall richness and diversity of the microbiome. And it's actually not just the bacteria that were in those foods, but lots of other bacteria as well. So kind of the overall health of the ecosystem. Now, this is in contrast to probiotic pills. So about 31% of our patients in our study were taking over-the-counter probiotics. And even when we eliminate those patients that were taking it because of antibiotic use, we find that those patients that were taking probiotics actually had a trend toward worse outcomes with immunotherapy. Now, again, correlation, not causation. So we go back into a mouse model. And here at the bottom, what we see is that those mice that we gave them these over-the-counter probiotics, we just went down to the local pharmacy to get, actually have worsened tumor growth and decreased response to immunotherapy. Now, probably a much stronger effect is actually on antibiotics. So there have now been multiple studies showing that antibiotic use within 30 days of initiation of immunotherapy is associated with a decreased response to immunotherapy. And then this was actually at the bottom, this was a patient that had gotten a fecal microbiota transplant, had a tumor response, and then was very sick and hospitalized and needed antibiotics. And obviously, when we need antibiotics, you need antibiotics. So patient got broad spectrum antibiotics, their tumor started growing again, and then they actually gave them a second fecal microbiota transplant and re-induced response. Let's skip that one. So conclusions, there is an intimate relationship between the gut microbiome and our immune system. There are distinct profiles between responders and non-responders to immunotherapy. But again, this is not one-to-one, and these are not tests that are clinically indicated yet. The microbiome can be intervened upon. What can you do overall to improve your gut health? Again, knowing that we are still working on those studies showing that this actually impacts outcomes. But so prebiotic foods, so basically things that the bacteria eat. So those are legumes, grains, vegetables, and fruits. Avoid processed foods, probiotic foods, but not probiotic pills. And then avoid unnecessary antibiotic exposure. And of course, there's still a lot of ongoing research as well, looking at things such as exercise in this context. And then just a couple of resources. So of course, AMET Melanoma has some good stuff out there about diet and exercise during cancer, during melanoma treatment specifically. AICR, which is American Institute for Cancer Research. Cancer.org, which is the American Cancer Society, and then actually a book that my colleague at MD Anderson wrote. I don't like the title. It's called Anti-Cancer Living, but it basically talks about diet, exercise, stress, sleep, social support. These are all things, right, that it's true. They are anti-cancer. I do just want to emphasize, though, that all of these changes and all of these lifestyle factors that we're talking about, this is really about trying to do everything we can to maximize the chance of response. Some of us do everything right and we still get cancer, or we do everything right and we still, tumors don't respond to immunotherapy or to targeted therapy. And so this is really just about, are there things that we can do to do everything that we can to improve outcomes? So thank you very much again for having me. I'm sorry I couldn't be there in person.